Our topic now is HIV/AIDS. This obviously is an acquired immunodeficiency. And truly, I've been saying to students for many, many years, if you understand what HIV does to the immune system, then conversely, you understand how the immune system is built. Because much as I named my central diagram of the immune system nirvana, in Hindu belief, the opposite of nirvana, in other words, immunological hell, would be narak. And truly, this is the pathogen that is the quintessential destroyer of your entire immune response. So understanding the system means you will understand the problem with HIV. Now, there's really nothing about this virus that you should not be familiar with. So I'm going to do both the life cycle of the virus and the immunological issues of how it causes destruction. That means when we come to our discussion of virology, I will have a gap there because of the fact that the only other retrovirus that we'll need to talk about is the oncogenic retrovirus that causes HTLV, leukemia. So this is a retrovirus family. That means it is a single-stranded positive sense RNA virus. It carries 2 copies of its genome. In other words, it's diploid. And the rationale for that is because it intends to actually parasitize the chromosomes of a diploid creature, namely the human being. And so if it only came with 1 copy of its genome to integrate into the chromosomes, it would mean that it had a less likely chance of actually causing a productive infection. Because remember, on average, we express the products of 1 chromosome of a set, usually not both. So it would be possible for the virus to integrate itself into an area that was not transcriptionally active. And if that were the case, that would be the end of the infection cycle for that particular sequence of DNA. Now, it carries with it an encoded reverse transcriptase, we'll talk about this more, an integrase to integrate itself into the chromosomes, and protease enzymes. We call it lymphotropic because of the fact that it infects lymphocytes, and specifically those that wear CD4. So that means it's going to infect the general of your immunologic army. I think you can see immediately that this is not going to be a pretty picture. It uses chemokine co-receptors to target other cells that wear CD4 leaflets as well, early on using the CCR5 chemokine co-receptor of the macrophage, and at that period of time we say that it is macrophage tropic or M-tropic. Later on, it switches over to targeting T helper cells using the CXCR4 chemokine receptor, and now we say that it is T-tropic. When you look at the virus itself, which I've drawn here, realize that the outermost materials are products of the envelope gene region in the genome of the virus. GP120 is what binds to CD4 and the chemokine receptors. GP41 is the fusion protein, which basically acts to fuse the membrane of the virus to the membrane of the host cell. And as we'll speak more about later, this is what makes it a syncytial virus and causes it to be especially important that a cell-mediated immune response be expressed against it. Because remember, syncytia, or multinucleated giant cells, are formed when viruses move from one cell to another by fusing cell membranes. And what that means is that there is not a lot of time within the life cycle of the virus that I could actually coat it with antibodies and block the binding to the next generation of cells. The group-specific antigen gene products are internal to those envelope gene products P17 being the matrix protein, which gives the virus its shape. P24, the capsid protein, which looks sort of like a truncated cone. This is the first molecule against which I will mount an antibody response. And notice these are all P for protein. I should have mentioned GP up above stands for glycoprotein. So the name is telling you both the molecular material that the product is made out of, as well as the molecular weight. 41 means 41,000. Now, P7, P9 are the molecules that are strung like beads along the diploid RNA. Those are the nucleocapsid proteins. The products of the polymerase gene region are the things which carry out the life cycle of the virus. The reverse transcriptase, which is going to take an RNA message and reverse transcribe it into DNA form. The integrase, which is going to be responsible for integrating that message into the chromosomes, and the protease gene product, which is responsible for cleaving the two envelope glycoproteins apart from one another. They are actually synthesized as a single large molecular weight structure, and with the protease enzyme activity, they are cut into unequal parts, allowing them to take the necessary position they should on the surface of the virion. Now, I'm sure you've all heard of protease inhibition as a mechanism that we use in antiretroviral therapy, and realize seeing that and how it would happen to change the appearance and the ability of the next generation of virions to adhere is just easiest when we're talking about the diagram of the anatomy of the virus itself. Epidemiologically, tremendous numbers of ways to be transmitted, basically intimate contact sexually or otherwise, can be transferred across the placenta in breast milk perinatally. In the United States at the current time, most of our new cases are happening in females or IV drug abusers, which means that it has taken a different epidemiologic route nowadays over what used to happen in the beginning of our discovery of the life cycle. The life cycle begins when the virus binds to CD4 and the chemokine receptors using its GP120 molecule. And I mention here something that you may well want to watch in your spare time. It's a very nice animation done by a German animator. There are actually 3 versions at this site on YouTube. And notice it's spelled with a K, replication, because that's the way it would be spelled in German. But there's one with English narration, there's one with German narration, and there's another one with some very emphatic and fun music to listen to if you'd like. But it's scientifically accurate and shows both the life cycle of the virus and the places at which the drugs would interrupt the life cycle. So I think you would find it beneficial to watch. Now, when we talk about the tropism of the virus, that's simply a fancy word for what cell is infected. And because of the fact that GP120 is subject to very frequent mutational change or genetic drift, it's true that we can have changes in the binding of the virus to cells that causes changes in the necessity for an ultimate immune response. The fusion protein, as we said, is what causes the formation of syncytia, and the drug that blocks that is enfuvirtide. Once the virus enters the cell, it then uses its reverse transcriptase to create a DNA version of itself, and realize that it is the reverse transcriptase which is really responsible for the wide amount of genetic drift that we see. because it's so extremely error-prone, it's almost constantly changing the format of the molecules that it is creating. And as a result of that, you have very dramatic changes in the need for a new immune response to be mounted almost constantly. The virus has a gene product called VIF, which is responsible for stopping the normal proteasomal degradation pathway that would take care of things found in the cytosol. Next, the integrase enzyme will cleave the ends off of the virion structure and will then create the DNA transcript, the DNA, starting again. The integrase enzyme clips off the long terminal repeats from the DNA that is newly formulated, and then circularizes that molecule and carries it in the form of a provirus into the cell. Raltegravir is the agent that we use to block that integration stage. But notice this is unique to anything that we'll talk about with any other viruses, the physical parasitism of the chromosomes as a function of the life cycle. The VPR gene product of the virus is responsible for making the nuclear entry possible and arresting the cell division process long enough for the virus to be integrated. The proviral DNA now, from its position inside the chromosome, is simply transcribed and translated eventually by cellular enzymes. So first, the creation of messenger RNA. The TAT gene product of the virus activates the production of that step. And then those molecules are transported to the cytoplasm where they're going to be translated into protein. And that step is controlled by the viral REV gene product. Those spliced messenger RNAs are translated on ribosomes and cleaved by the protease enzyme, which we mentioned before. And anything that isn't spliced is going to be packaged as a copy of the genome, because remember, it comes and goes in the form of single-stranded positive sense RNA. The assembly is under the control of the VPU gene product, and eventually the virions are released by budding or by simple direct transfer from cell to cell through the membrane of the fused cells. Now, if we look at the course of an untreated infection in a person with HIV infection, realize the mean time to death in untreated cases would be about 10 years. And it is universally fatal because of the destruction of the immune response. But the best way to calculate what the process is happening in the patient is to look at the number of CD4 cells that are persisting as you go. The normal value of CD4 cells is about 1,000 per microliter of blood. And that, of course, will take a nearly linear decline through the course of an untreated infection for a number of different reasons, one of which is that the virus will actually cause cytopathic effects in those T cells. But initially, there will be an immune response against the virus. Notice here the blue line signifying the increase of cytotoxic killer cells, which would be necessary to hunt down and kill virus-infected cells. But I have to keep in the back of my mind here that that very killing is also contributing to the decline in number of CD4 cells, because the cells who are infected are my CD4 cells. So your immune response actually contributes to the problem. The red line here signifies the virus production, And of course, that early spike would be getting the virus into as many cells as possible, first infecting every macrophage it can find, and then later on spilling over into the infection of T helper cells. I will make antibodies against the various viral products. As I said, the first one made would be the antibody against P24, the capsid antigen. And notice that that antibody is going to decline late in infection due to the fact that it is mostly of IgG isotype. Now, as always, it would be IgM early on and then isotype switch. But remember, IgG is an antibody that requires T cell help. So there's going to be a point in the progression of disease where I'm not going to have enough T helper cells anymore to mediate that isotype switching. All right, now, as I fall lower than that 1,000 value, most patients will remain generally asymptomatic until they fall below 600. Okay, at 400, you would find that the patient now no longer has enough T helper cells for isotype switching. So that is the point at which the production of the P24 IgG antibody declines. But notice the IgM antibodies against the envelope antigens, GP41 and GP120, those will remain elevated until death because they are IgM in isotype, and also because they are subject to tremendous genetic drift of the molecule. So the patient is constantly making new antibodies to reflect the changing appearance of the antigen. So this is a constant primary immune response with no T cell help. Finally, when the patient reaches 200 and below, this is the definition of AIDS, along with the presence of an AIDS-defining condition, because really at this point, the immune response totally collapses. There's not enough of any immune response to protect you from opportunistic pathogens. The virus returns into the bloodstream, creating viremia, even though it has been kept at a low level up until this point by the presence of antibodies which caused the virus to be removed from the blood. But now the virus returns into the blood, the viremia goes up to astronomical amounts, and the patient succumbs of a total absence of any immune response whatsoever. Now, what comes beneath is simply exactly the same material in verbal form. Since I've already walked you through it, I will let you read those on your own. But let's talk more now about the immunopathology of this disease. Because remember, as we were building the system before, I said to you that my APCs all have CD4 on their surface. My T helpers all have CD4 on their surface. So what's going to happen to the Nirvana diagram? I'm going to kill the T helpers first, certainly, and there is an immunological deviation early on that causes me to kill the T helper 1s faster than the T helper 2s. And that's one of the reasons why the antibody production will persist longer than you would anticipate. But still, eventually, I'm going to end up killing all of those cells which I described as the generals of my immunologic army. I'm also eventually going to lose the functionality of my antigen-presenting cells, although they tend to be much more resistant to the cytopathic effects of the virus. They still are not operating normally because of the presence of the virus and the fact that there's no one to signal downstream. So if I have cut the head off of the Nirvana diagram, realize that down below at the effector level, I really cannot do anything in a concerted way because I'm getting no orders from the generals of the immune response that would necessarily be delivered to the soldiers of cell-mediated versus humoral immunity. Now, in addition to that, we've got a tremendous number of other indirect mechanisms that we can talk about. One of the things that I didn't mention is the fact that the activation of cells is going to release more virions out of that cell. And remember, your generals, their most important function is to clone themselves. If you recall the Rubush rule of twos, that they will make the cytokines that will cause themselves and all of the effectors beneath them to clone themselves. That very act of cellular activation also replicates more viral genetic material as well as the DNA of the cell itself. So there's really no mechanism by which I could turn that off when I need those cells multiplying in order to respond to daily challenges in my life. In addition to this, the budding of the virus causes a loss of integrity of the membrane, so the cells will die faster than you would anticipate. Unintegrated viral DNA accumulating both inside and outside of cells ends up causing problems of direct toxicity. Impaired RNA processing is going to slow down the ability of the cell to do the things it naturally needs to do. And as we said, syncytia formation is going to make the virus difficult to get rid of by antibody synthesis. I will cause defects in intracellular killing as a result, intracellular signaling as a result of the presence of the virus. And since all signals come from the general, all things downstream in the nirvana diagram are going to be shut down. Cells will undergo apoptosis more frequently than they will. And I even have a depression of the production of lymphocytes out of the primary lymphoid organs. Activated cells will undergo activation-induced cell death, the trimerization of Fas and Fas ligand. And really centrally, I probably should have put this much higher up on the list, but remember that anything that you do immunologically that would be valid in any other viral situation is going to kill the very cells you need to continue your life. There truly is no answer to this problem built into the immune response. And so it's the reason why it's so important for you to understand this pathogen, because it is simply the opposite of what you want to occur in a normal immune response. This is one of those viruses that will shut down MHC class I expression. That is done by the expression and production of the NEF gene product. But I should say to you, it might not be intuitively obvious why it would be advantageous for the virus to shut down the expression of CD4 molecules on cells. Well, realize that's the molecule it needs to attach to, so that sort of seems counterintuitive. But if you think about the idea that once a virion gets into one of your CD4 cells, it's basically sort of hanging an occupied sign on that cell. Okay, sort of like going into the bathroom and turning the latch and then it says, no, somebody's in that bathroom stall. Well, it is telling the remaining virions out there, don't take this cell, I've already got it occupied. And so that's what contributes to its spread so dramatically through the body, because it tends not to reinfect cells that already have virions going through their life cycle inside. Now, when we talk about the advent of opportunistic disease at the point that the patient falls to 200 CD4 cells and below, Remember, that is the definition of AIDS as far as the cell count is concerned. But all of the things on this list are AIDS-defining conditions in the manifestation in which they're described. And although I'm not going to read this to you, and although I don't expect you to memorize individual numbers here, notice the trend that I tried to show you in the color system on the right-hand side column. The initial problem is that the virus kills Th1 cells preferentially over anybody else. So I will end up limping along with T helper 2 cells still working, but those cells are causing the production of antibodies, and there's really been no evidence that antibodies help the patient at all. So my earliest symptoms are from the problem of those failed T helper cells. Then I lose the ability to protect my barrier systems, my skin and my mucous membranes. I then have breakdown of the blood-brain barrier and the invasion of the central nervous system. I have the production of cytokines that cause the wasting and weight loss and fatigue, primarily tumor necrosis factor, and eventually, of course, systemic collapse of all of the systems of protection of the body. So you can predict the sequence in which things will happen. And you should notice, I think, that toxoplasma is the #1 diagnosis at autopsy in HIV patients in the United States at the present time. That encephalitis and the formation of those lesions in the brain that are described as ring-enhancing lesions. Diagnosis of HIV has become much more elevated in its ability in recent time. We now start with an antigen-antibody immunoassay that will detect the 2 major clades that we have in the United States, and then a conformational differentiation technique, which would allow me to tell the difference between the 2 clades. Okay? We do those both by antibody differentiation and by nucleic acid testing in the United States. If you want to look at the viral load of the patient, in other words, what is the level of viremia, realize this is what is usually used as our management technique in order to tell how a patient is responding to antiretroviral therapies. That is done by RT-PCR. You're actually detecting the RNA of the virus in the blood. But remember, none of these earlier techniques would work per se in a newborn. So what I have to do in that case is use PCR to detect the DNA of the virus integrated into the host cells. Okay? I'm not going to be able to use an antibody assay because the antibodies are likely to have come from the mother. And so I have to look for the DNA, the integrated proviral DNA, because of the fact that even the viral load will be insignificantly small in a newborn from a mother who is infected. So make certain you know how to diagnose the infection in a newborn. That is a very, very common question. We use P24 antigen as a measurement of early infection, the early marker of exposure and infection, and realize that the ratio of CD4 to CD8 cells is actually something that should, in a normal patient, be about 2 to 1. In other words, every cell in the blood that's a lymphocyte, I have twice as many T helpers as I have cytotoxic cells. So my ratio of helpers to killers is normally 2 to 1. Once I reach the point that the patient has fallen to around 400 of those cells remaining, my ratio has become flat. And when I have moved to the point of being 200 and fewer, my ratio is now inverted. Now, that is a much more gross measurement, but it still is monitored in persons positive for HIV. It's just that I'm going to watch my patient, as you'll see, by trying to keep the viral load as low as possible. And I'm going to assume that by so doing, I'm going to keep that patient at a more normal level of the helper to killer ratio. Management, as I said, we follow that CD4 count, and we follow by RT-PCR for the viral load. Okay, antiretroviral therapy is going to be necessary for life, and the cocktail is 2 nucleoside reverse transcriptase inhibitors or non-nucleoside reverse transcriptase inhibitors and a protease inhibitor. We'll talk about the drugs more later. If anything happens that causes the viral load detection to start to increase, then that generally is a failure, an indication that I need to change the cocktail because the virus is developing resistance to that set of drugs that I've been using. Now, for the antiretrovirals, this has been a tremendous success story over the last few decades since HIV was first described. And we now have a proliferation of molecules that we can use to improve the lifespan and the health of persons who are positive with HIV. Maraviroc, if you notice, has in its name that it is a macrophage receptor antagonist. It is a blocker of CCR5. Well, remember, that's only going to work very early in infection because later in infection, the virus switches over to using the CXCR4 receptor instead. Enfuvirtide has something about fusion in the name. That's the blocker of the fusion protein, GP41. Raltegravir has portion of integrase in its name, so it blocks the integration. And then any of the drug names that are anavir, enavir, inavir, or onavir, those are all protease inhibitors that will block the creation of the appropriate sized molecules after translation. We have a huge number of nucleoside transcriptase inhibitors and non-nucleoside. Notice most of them have INE as their last 3 letters, but there is a proliferation of those, and I think knowing which would be used is probably beyond the scope of step 1. The prophylactic drugs that are used against opportunistic infections are asked. So make certain that you know the level of CD4 count at which I would institute particular protective therapies. When the patient falls below 200, I begin therapy against pneumocystis with trimethoprim sulfamethoxazole. Easy for me to say. As they fall down below 100, notice I add on therapy against histoplasma and toxoplasma, okay, itraconazole, and adjusting the dosage of trimethoprim sulfamethoxazole. Notice with histoplasma, we'll talk about the fungi in another session, but the endemic area, remember, follows the drainages of the Great Lakes straight down the Mississippi, Missouri, and Ohio riverbeds to the Gulf of Mexico. So would not be instituted in California, would not be instituted in Florida, okay? Cytomegalovirus is therapy induced at less than 50, along with Mycobacterium avium intracellulare and Cryptococcus, all three of those, in my opinion, you can assume, even if they do not mention this on an exam question, if they're giving you a patient with cytomegalovirus, disseminated infection, Mycobacterium avium intracellulare, or cryptococcal meningitis, they don't actually need to tell you that that is an AIDS patient. Because if you notice, this is a reflection of the fact that the immune response is basically gone at this point. And so adding on those drugs, I'm now basically throwing the entire kitchen sink at this patient. Ganciclovir, azithromycin, fluconazole added on to everything I had up above. Okay. Cryptosporidium is one that I held off on, although it's in that same group. Honestly, we don't have good drugs against cryptosporidium. And so you'll find that a patient in end-stage AIDS syndrome will basically die with diarrhea, although it may not be because of the diarrhea that they actually die. It's very difficult to manage. It's a chronic, unrelenting diarrhea, and the only thing we do have is nitazoxanide, and it's just not terribly effective. Prevention, we now can talk about PrEP, the pre-exposure prophylaxis, which cuts down infection 99% in sexual transmission, 74% in IV drug users. So a tremendous advancement. The trade name Truvada is the one that contains tenofovir, and that's the one that inhibits the reverse transcriptase, and then encitrabine, which is a cytidine analog. So that combination is used in the pre-exposure prophylaxis. Of course, we do blood and organ donor screening. Safe sex, always a better idea. But it is truly a miracle that we have achieved what we have with this virus over the decades that it has, since it has first been discovered. So I did not do a hashtag MMR for this chapter. I hope you see why not. If I have to explain to anyone why should you care about HIV's life cycle and prevention and therapy and how it destroys the immune response, then you really haven't understood very much about the medicine of the human immune response. So that seemed sort of like beating a dead horse. I do want you to make sure before you leave this chapter, when you look at it on your own, that you can answer these questions for yourself as usual. And remember that almost any question about how the immune response works can be posed in the context of an HIV positive individual, and then you will be answering the question, how doesn't it work in that scenario? Terribly important pathogen for you to understand.